Parasitic diseases of the central nervous system. A general overview of common neurological parasites highlighting some recent toxoplasmosis research. Parasitic diseases of the central nervous system can be life-altering. Delicate neural networks maintain local homeostasis. However, several pathogens are capable of entering the central nervous system. Infection can result in morbidity and mortality. Anyone, anywhere can be infected by a parasitic disease, although those living in areas with limited resources are at a greater risk. Those with compromised immune systems are highly susceptible to debilitating consequences once infected. Neurological sequelae can affect mobility, sensory organs, cognitive functions, and even cause seizures and epilepsy. People suffering with the diseases have lifelong disabilities, increased poverty, and face social stigma. Parasites can be classified into prokaryotic unicellular organisms or multicelled helminths. Prokaryotes have a great ability to multiply in the immunosuppressed, and helminths cause physical disruption to the host tissues. Some of the more common parasite infections of the human central nervous system are cystic cirrhosis, toxoplasmosis, echinococcus, and malaria. These diseases have diverse causes, vectors, intermediate hosts, modes of transmission, and geographic distribution. Presentation of disease often exhibits crossover signs and symptoms, making it difficult to diagnose. Almost all are associated with seizures and epilepsy. Some cause eosinophilia, characterized by eosinophils in the cerebral spinal fluid. Eosinophilia in the cerebral spinal fluid has been arbitrarily defined by counts greater than 10 eosinophils per milliliter. They play an important part in immune response, but can cause irreparable tissue damage to the infection site. Most commonly caused by helminthic infections, but may also be caused by other types of infection, thus making differential diagnosis a challenge. The blood-brain barrier and blood-cerebral spinal fluid barrier maintain homeostatic neural environments. Endothelial cells, astrocytes, and neurons, along with the extracellular matrix, make up the barrier. The choroid plexus functions as an interface between the blood and cerebral spinal fluid. Protozoan and helminth parasites can infiltrate the central nervous system by either crossing or disrupting the blood-brain barrier or the blood-cerebral spinal fluid barrier, evading immune response. The tight junctioned barriers are selectively permeable to macromolecules and hydrophilic molecules. In certain cases, such as Plasmodium causing malaria, the blood-brain barrier is broken down from hypoperfusion and hypooxygenation. The parasites can achieve that by modifying the host's red blood cells. There are other mechanisms of blood-brain barrier disruption not well understood, but are thought to involve areas such as the choroid plexus that do not have tight gap junctions. Other crossing methods are thought to be done by a coevolution relationship between pathogens and hosts, almost as though they've developed a strategy to deal with the central nervous system's specialized immune response. Host factors thought to be significant in parasite exploitation are hormones, cytokines, neuropeptides, chemokines, and red blood cells. A silent invasion leads to a chronic infection which can translate significantly to morbidity and mortality. Toxoplasmosis. Toxoplasma gondii are intracellular parasites that can infect almost any nucleated mammalian cell. They exist as oocysts, tachyzoids, and bradyzoids. Tissue cysts in rodents and birds are ingested by cats where sexual reproduction of the T. gondii takes place in the stomach. Fecal oocysts are passed, then contaminate a cat litter or soil containing oocysts or undercooked meat, contaminated vegetables, can all cause the tachyzoids to be taken up by the host. They will then multiply by asexual reproduction through the host's cells. Organs or blood transfusion from an infected host could also cause infection. Tachyzoids are transferred transplacentally from mother to fetus. Toxoplasmosis is a global health issue as 30 to 50% of the human population is infected. It will disrupt fetal brain development, severely affects brain of host, linked to a multitude of neurological disorders, changes host cells' gene expression, modulates behavior and personality traits like anxiety, alters spatial memory, altered recognition of social novelty. It's linked to an increased risk of traffic accidents, and it's also associated with significantly reducing gray matter density in parts of the brain in infected schizophrenia patients. It doesn't seem to be the cause of schizophrenia, but could certainly increase the risk. Tachyzoids exhibit high metabolic rate and tremendous demand for nutrients. When present, there is constant demand for an energy and biomass supply. They can remodel the host's intracellular processes to gain access to the central nervous system. Taxozoids live in vacuoles resistant to lysosome fusion. Avoiding degradation, they spread through the bloodstream and infect monocytes, dendritic cells, neutrophils, macrophages, and lymphocytes, thus gaining access to the tissues. This is known as a Trojan horse mechanism. They can also directly branch intracellular junctions and transmigrate between cells. Once inside the central nervous system, the T. gondii dispense and invade astrocytes, microglia, and neurons. After invasion, replication drops, bradyzoites persist as a latent infection for the host's lifespan. T. gondii secrete effector proteins that can modulate host cell gene expression, altering transcriptions for glycolysis, cholesterol biosynthesis, carbohydrate and lipid metabolism, transcriptional regulation, protein synthesis, cell cycles, apoptosis, cell signaling, inflation, and cell adhesion, effectively altering the phenotype of the infected cell. Maintaining a constant pyruvate supply has been shown to be critical for asexual reproduction of toxoplasmosis. A T. gondii infection causes an increase in glycolysis for the host's infected cells. Host metabolism is profoundly affected in a way similar to that of the Warburg effect of patients with certain types of cancer. Normally, in oxygenated cells, energy production is most effective through oxidative phosphorylation. T. gondii causes host cells to switch to aerobic glycolysis, leading to an increase in production of the end product of glycolysis, pyruvate. Pyruvate is critical for human metabolism. And pyruvate metabolism plays an important role in neurodegeneration. It's critical for mitochondrial ATP generation and for driving several major biosynthetic pathways intersecting the citric acid cycle. Once the pyruvate is in the mitochondrial matrix, it can be converted to acetyl-CoA. Pyruvate dehydrogenase complex, PDH, converts pyruvate and NAD+ into acetyl-CoA, NADH, and carbon dioxide. Acetyl-CoA generates acetylcholine, a key neurotransmitter. Citrate and malate are intermediates of the Krebs cycle. They are involved in a variety of biosynthetic processes and build up with a reduction of ATP, with a reduction of the ATP producing cycle. Decreased ATP production will severely affect tissues with a high demand for that energy molecule. Altered pyruvate metabolism is found in several neurodegenerative disorders. Disruption of pyruvate metabolism is associated with Alzheimer's disease. That includes problems with memory and behavior, synaptic hyperexcitability, increased production of amyloid beta peptide, which is widely accepted as the main pathogen in Alzheimer's disease. Parkinson's disease, which involves mitochondrial dysfunction, loss of dopamine neurons leading to significant motor and non-motor neurological dysfunctions. Genes regulating pyruvate metabolism are underexpressed in Parkinson's disease. Leigh syndrome. People with Leigh syndrome are less likely to interact with the environment. They respond slower to sensory stimuli. Primarily affects the basal ganglia, thalamus, and brainstem. Shortly after T. gondii actively invades host cells, dendritic cells exhibit enhanced GABA synthesis and infected cells secrete GABA. Brain-resident immune cells, such as microglia, and circulating immune cells, such as neutrophils, monocytes, macrophages, and dendrite cells, are thought to secrete GABA and/or express GABA receptors and associated proteins. GABA is commonly known as an inhibitory neurotransmitter, can be depolarizing or hyperpolarizing depending on chloride concentration. It has roles in other cellular processes, such as migration, proliferation, differentiation, synapses formation, axon growth, and neural death. It's worth mentioning that there was a delocalization in a toxoplasma-infected rodent brain of the enzyme GAD67, which is used in GABA synthesis. Such a finding indicates disturbed GABA signaling. Infected rodent brains also produce a greater expression of AVP neurons and neuropeptides in the amygdala, thought to be the main regulators of reproductive and social behaviors. Greater expression on AVP neurons is also associated with loss of fear. Dopamine pathways greatly implicated in neurological disorders associated with chronic T. gondii infection. Disruption of non-coding RNA MIR132 involved in differentiation of dopamine neurons appears in all types of Toxoplasma gondii strains, with upregulating in acute and downregulating in chronic infections. Biopterin is a coenzyme synthesized in several parts of the body. The catalytic activity of amino acid hydroxylase depends on biopterin, a coding sequence of the T. gondii genome. Fundamental to this process was remarkably similar to that sequence in the human genome. Parasites have a unique ability to modulate brain physiology. Extensive research is needed to better understand the mechanisms at play in order to uncover a possible approach to curing neurological disease. Wash your hands and fully cook your meat. Thank you.